So let me introduce Henk Britz. Henk Britz is a PhD student from the University of Pretoria. He's been studying African horse sickness virus VP2. People should know that African horse sickness is a major threat to the equine industry in southern Africa, and it's been a long time in coming that people have been able to study all the various proteins associated with African horse sickness virus. And Henk has made a major breakthrough. So let's hear about it, Henk.
Yeah, thank you for the introduction. Okay, so I'm actually a master's student, but yeah, hearing some echo. Okay. Anyway, so I'm going to talk about a bit of solving the molecular structure of African horse sickness VP2. Just a brief introduction. African horse sickness is caused by African horse sickness virus. It's an virus, which makes it closely related to bluetongue virus. It's spread by Collocoides midges, and it affects horses and other non-native equines, as the Dutch discovered when they brought horses to southern Africa, and they had a few major outbreaks, and the mortality rates are really quite high. It's something like 50% in the best of cases, and it can be as bad as the entire form, where the most severe form causes this frothy discharge from the nose, and it's really an awful disease. So there are some detection methods, traditional detection methods available. You can have ELISA kits available, and but it's a really aggressive disease, so PCR is actually much more appropriate. The only reliable prevention measure is a multivalent live vaccine. One of the problems with the vaccine is that you can't distinguish natural immunity, vaccinated immunity, and current infection. So this makes export of horses really a problem, because they will all just test positive whether or not they were vaccinated, whether they had previous infection, or if they're currently infected. You can't really tell the difference. There's also a reversion to virulence that can happen because the virus has separate RNA segments. They can reassort in the same way that influenza virus reassorts. And some of the vaccines have actually reassorted into a virulent version of the virus. So that's some issues with the vaccine that could be solved by reverse genetics. And to do some of this work, it will be useful to have a more complete map of the virus. So it's a non-enveloped virus. There's 10 segments of double-stranded RNA. It encodes for 7 structural and 5 non-structural proteins. And you can think of it as having 3 protein layers. There's a thin shell that makes up the inner core. Attached to that is VP1, the RNA-dependent RNA polymerase. Attached to that is VP7. They are trimers that form these little stalks that attaches to VP3. And then the outer capsid has VP5 and VP2. So it takes a T2 icosahedral arrangement. So there's a slight deviation in structure from T equals 1. And for this, it needs 2 conformations. So you've got an A conformation that 5 of which VP3s can attach to form a 5-fold vertex. And then the B conformation can form a 3-fold vertex next to the 5-fold vertex. And that structure just repeats around the virus. And then attached to this VP3 is a VP7 trimers. And it creates 13 unique attachment points. So for the outer core, it's a T equals 13 icosahedral arrangement. So a bit about how the structure and the function relates with each other. There's a small pore at the 5-fold vertex that can be used during RNA threading. And recently they've shown that this is actually part of the assembly model. There were sort of competing models. The one said RNA first assembles together and the core forms around it. The other model said no, the core forms first, the RNA then gets transported into the center of the core. But it was now shown that they're sort of synergistic. The RNA enhances the assembly and the assembled form enhances RNA packing. Okay, now moving to the outer capsid. This is where the receptor binding protein and the fusion protein sits. So VP5 is the fusion protein. It undergoes conformational change under pH conditions, low pH conditions in the endosome. And then that induces VP2, the receptor binding protein, to actually detach. And then this altered VP5 then is able to insert into the membrane, breaks it open, and then the core is released into the cytoplasm. So I focused on VP2, the receptor binding protein, and it's also the protein that most of the neutralizing antibodies bind, and it's also responsible for serospecificity. So for vaccine design, is quite an important protein. Previous work done on whole African horse sickness virus resolved VP2 up to about 14 angstrom. For bluetongue virus, they've resolved it to 3.6 angstrom. So I wanted to try and get closer to a resolution where you could perhaps model a VP2. Okay, so the objectives for my project was to obtain sort of in silico models to help with structure determination. So this is this new wave of deep learning-based structure prediction models that came out recently. Next was the production and purification of African horse sickness VP2, so try to crystallize it. Then was I wanted to do some X-ray crystallography or CryoEM to just determine the structure, and then I could compare bluetongue virus VP2 with African horse sickness VP2. So what I first did is I extracted some electron density from the whole virus structure. The tip domain, which is the top side that sort of sticks out here, was not resolved yet at this point in time. So I wanted to create a more complete structure. I saw the electron density was available, but you couldn't really manually model it. So I used Rosetta Fold. At the time, it was outcompeting AlphaFold 2 with certain large proteins. So I decided to use that, create a more complete structure of bluetongue virus VP2. It's also a good introduction to the different types of software I would probably be using. So this was a good exercise to get familiar, get a more complete structure of bluetongue VP2. And then, yeah, so using this Rosetta Fold generated structure, I nudged it into place into the actual electron density. I used Phoenix dock and rebuild as a starting point. So it will break your protein up into different domains. It will do rigid body refinement and then reconnect the components in a way that fits the backbone in the electron density. And that way I could get a more complete empirically verified structure for bluetongue virus VP2. And here's just the zoom in region of the zinc finger domain that was also modeled by the Rosetta Fold. It didn't add the zinc though. I had to add the zinc in manually, but you can do it partway through refinement. Okay, moving on to African horse sickness now. So I have a colleague at a company where I previously worked. He had a way for transient expression of problematic proteins. So he took a guinea pig codon optimized cDNA plasmid encoding for VP2 under a cytomegalovirus promoter. And then through transient expression, we were able to get soluble protein. And I used strep tag affinity chromatography to purify the protein. And as you can see, it's really quite pure. Didn't have to do any size exclusion or additional purification. And that was good because yields were really low. At best, I got less than 0.3 milligram per milliliter. Milligram total yield, not even per milliliter. So it didn't even have a milligram total yield. But it was enough to at least try some crystallization. And what I found was I got needle crystals, which can happen to proteins, but also found these hexagonal crystals, which seemed quite promising. I looked at some references of biotin crystals because I didn't do any buffer exchange, so the biotin in the elution buffer is actually still present. So looking at the hexagonal crystals, I had a good confidence that this was the actual protein, but our diffractometer was broken, so I couldn't do any screening. And by the time someone said I should just send it to Diamond anyway, the plate has dried up. I did produce more protein in the meantime, though, and this was close to the time when my supervisor forwarded an email to me that Jeremy Woodward at UCT was doing a course on CryoEM, and I thought, oh, maybe I can do CryoEM since my yields are so low. Okay, so went to Cape Town, and one of the first things you do is a quality control check. It's also a good way to familiarize yourself with the EM 3D reconstruction pipeline. So the first thing we did was some negative staining. This is where you take your protein and put it on a carbon-coated copper grid, add some electron-dense element, so here in this case, uranol acetate, and then that will block some electrons from passing through. And through negative staining and shadowing, you can indirectly look at your protein. You can even do some 3D reconstruction of that. And I was able to do a 23 angstrom reconstruction. So a little bit about the orientation problem that I ran into. You'll notice that the one image only showed the top view. That is because the protein has a bit of an affinity for the carbon, so you could only really see one view. We hope that this was only the case for negative staining because the carbon, the hydrophobic side of the protein might attach to the carbon, so in water it might not be a problem. But I still ran into some orientation problems. This view shows you the ideal versus a suboptimal condition. And here's a negative stain micrograph where you can see only the top view, and this is the same sort of image at a 60-degree tilt. And you can see you only really see the top view here. And on the right side, you can actually see multiple sides just because the axis of symmetry might be perpendicular to your initial view angle. So if you tilt it, just by rotation, you can see a whole bunch of different angles from the side. Okay, so the first step in 3D reconstruction is to pick your particles, and then you can classify them by how they look. So this can be how clean they look and what view angle the particle sort of belongs to. It can then generate these class averages, and then you can start cleaning them up a bit. So you pick the best looking classes, and then you can classify again, you repeat that until you're sort of happy with the class averages that you're getting. You can then use these class averages to generate a 3D reconstruction. So one of the first things that you need to do is you need an initial model, and your software will then generate a bunch of projections from this initial model, and then it can start assigning angles to all of these different particle classes that you have. And during 3D refinement, you start with a low resolution initial model. If it's not low resolution, you low pass filter it to prevent model bias. Generate some projections. How finely you generate those projections you can control through your software, but in general, you make a bunch of coarse projections, and then most of the actual comparison work and reconstruction work is actually done in a reciprocal space. So you can get a Fourier transform of your projections and a Fourier transform of your particle in question. You can start comparing them and then figure out where they sit in space. And then once you have all of these particles assigned and aligned with different projections, you can interpolate by filling in the gaps in between. And then from this 3D reciprocal space, you then do an inverse Fourier transform, generate an improved model from which you get improved projections. And this is how the refinement then begins to cycle. And you get better and better projections and then better alignments until you reach a point where you don't see any further improvement. Okay, so moving on from negative staining to CryoEM. What you need to do is you need to vitrify your samples in order to view them through transmission microscopy, because any crystals ice crystals will mess with the way that the electrons scatter. So you just want a glassy ice in which your protein or your virus or your complex is suspended. And to do this, you need to freeze it rapidly and inside of liquid ethane. So you have a dewar that you cool with liquid nitrogen. Once it's cryogenic temperatures, you pump in some liquid ethane, or rather you pump in gaseous ethane that will become liquid ethane. Then using something like a vitrobot, you can rapidly plunge in your grid that contains your sample, and then all of this needs to be kept at cryogenic temperatures while you're using the microscope, transporting the sample. All of that needs to stay frozen, and you don't want any crystalline ice. Okay, so now that we've moved to CryoEM, we run into some contrast problems. You don't have negative stain, so you need to get contrast through other means. So what you do is you look at the sample out of focus, and how much the more you increase the defocus, the more contrast you get, but you lose some information in the process. It's a bit of a trade-off. You also run into an issue where the contrast contribution begins to oscillate. And this happens because when you defocus, you increase the amount of phase contrast. And that is what gives you the enhanced contrast from defocus. And you get this constructive and destructive interference of electrons because of the different path lengths that they travel. And the inherent process of scattering also causes a phase shift in the electrons. And all of that causes these sort of gaps in your information that you get from these images. So what you need to do is you can actually map the contrast transfer function. It gives you an idea of how your image is perturbed by physics and by the microscope. So you can get an idea of how your contrast will oscillate, and you also get an envelope function where, as you approach higher spatial frequency, you start losing more and more information due to the nature of your microscope. So this is due to spatial and temporal incoherence. So when electrons have too much energy, you lose them because you have to energy filter them, and you also have spatial incoherence. So that's sort of a blurring, which means that the high frequency elements will start getting blurred away. So you get this squashing of information at high spatial frequency. So these things sort of limit CryoEM resolutions. So what you can do is you can map the contrast transfer function to your power spectrum. So you take your images, you do a Fourier transform, and then if you look at something close to focus, you get a particular pattern, and then you get a more squashed pattern when you're further from focus. So these will have different defocus values and have a different contrast transfer function. So it's important to have an accurate estimation of defocus and to have a good contrast transfer function to be able to extract the most amount of information from your images and get to those higher resolution particles and eventually reconstructions. So this is an example of how a contrast transfer function can fit your power spectrum. And using this, you can see how you can fix your information to look more like an ideal case where you had a perfect microscope and you were able to account for the way that the electrons interfere with each other. But because I had the orientation problem where my particle was just sitting flat even in ice, what you get is when you tilt your stage, one part of the image, so that would be the blue block, will be closer to focus than the other side of your image. So just because they're physically closer, they're also closer in focus. You also get more beam-induced motion from these tilts. So you get a perceived motion in line with your detector. So it would look like everything is moving due to the beam. We can fix beam-induced motion and motion from temperature differences through motion correction. But in order to fix the defocus estimation issue, you need to use a different approach. So what I end up doing is I cut my image up using a simple Python script. And the idea was to get a smaller zone. You reduce the amount of information used to get your contrast transfer function, but your defocus estimation will be much more accurate. Another drawback is the smaller the zone, the more your particles might accidentally be cut up and it's split between two contrast transfer function zones. So it means you kind of lose that particle. So I decided to just test a 1000 by 1000 pixel image splitting method. And that actually gave me a pretty decent result. So as you can see here, due to the tilt of the image, your defocus can actually vary quite a lot. So it's important if you're working with split images to not just trust that it's only going to be close to accurate for maybe a center piece of your image and the rest will be off. So it looks a bit complicated, but this is the full pipeline that I used. And I used two different particle picking methods. So for the first approach where I used the whole image, I had to use a Laplacian of Gaussian autopicker. This just looks at blobs, so it doesn't really look at anything with much detail. It just looks at a blob that might be a protein and says that's a particle. Then you can filter out the junk later. Versus reference-based picking, which I used in my split image approach, because reference-based picking didn't work with bad CTF estimations, with bad defocus estimations. So with my split image approach, I was able to use a reference-based picking method. And it probably has something to do with the way that Relion compares the Fourier transformed images. If you have a bad CTF estimation, you might not be able to resolve all of the detail. But if you have a Laplacian of Gaussian autopicker, it just looks at blobs. It doesn't do anything much more sophisticated than that. So with my whole image approach, I got a 4.3 angstrom reconstruction, which was not bad, but the first reconstruction looked not good. And so with my split image approach, I was able to get a 0.3 angstrom improve looks like for my reconstruction. So with inaccurate defocus estimations, you can see that you can't resolve nearly as much information as something that has an input with CTF estimations being much more accurate early on. I then decided to take the split image version and see if I can maybe model. It was difficult to do manual modeling, but at the time AlphaFold 3 was released. So I modeled this orange, this orange AlphaFold 3 output for African horse sickness serotype 4. And it seemed to match pretty close, but there's still uh, some loops that don't fit really well. So I decided to do some manual refinement in Qt, and then I did a few iterations of Phoenix refinement, sort of going back and forth, uh, improving the model parameters. And this is sort of what my final model looks like. It's about 70% of the model, so the tip domain unfortunately wasn't resolved, and there's a few reasons for this. It's worth noting that the sample was 6 months old before at the time of freezing, so it's luckily the protein was really quite stable. Um, so that some instability in the tip domain might be the cause of the problem. There's also some inherent flexibility if you can trust the AlphaFold 3 model. So the uh, tip domains might only be properly fitting into place once it's attached to the virus for whatever reason. And the lack of orientation, so more side views of the model might also have helped a lot in solving the tip domain. And yeah, so now that I've modeled the African horse sickness virus VP2, I could then compare it to bluetongue virus VP2, and I noticed two different insertions. So there's the helix loop helix in the African horse sickness in orange, and then for bluetongue virus, there's this hairpin loop, and they're sort of respectively unique to each version of the protein. So that's something to look into for the future because this is likely alternative ways that they interact with VP5. If you recall from earlier, VP5 is responsible for VP2 detachment. So this might be different ways in which VP5 pushes VP2 off of the virus. I then revised the sequence alignment now that I have structural information, so I could look at how far the residues are from each other in space and what secondary structure assignments they had, rather than just sequence identity alone. Sequence identity alone can be a bit misleading. The most parsimonious answer is not always the true answer. So if you have a structure, you might be able to see what parts moved around where, and yeah, sequence identity was really quite low, even after making some adjustments. It's only 13%, but it shows how close the structure can be despite low sequence identity. There's this purifying selection for structure, not just sequence identity, that keeps them, at least where they interact with VP5 and VP7, there's some constraints in how the protein can actually look in those regions. So where the tip domain exists, there's the least sequence identity, and closer to the trimerization domain, closer to where it interacts with VP7, the sequence identity is a bit higher. Okay, some acknowledgements. This work wasn't possible without the funding from the Chang Zuckerberg Initiative, a lot of assistance from EM, Electron Microscopy Unit at UCT, and then Deltamune, who funded the protein synthesis for this project. And thank you very much to EBIC for giving us some beam time to collect some images.
Thank you very much, Henk. That was a very interesting talk. And I'm so pleased that you actually went through everything in detail and showed people how the whole process is done. I think that the audience is probably appreciating that. It's all very well for them to see end products and how everything figures out beautifully, but to understand the struggle is something different. Does anybody have questions for Henk? Are there questions? Is anybody putting up their hand? Okay.
I don't see any, but I
Okay.
have just one.
Yeah.
How would you test this the theory, I guess, that the flexibility is a feature in the protein binding with the virus particle, or in the virus particle? Is it possible to I guess you're just doing the protein, but not the entire virus particle. Can you take data of the entire virus particle?
So it's a bit difficult to get all of the approvals necessary to get a full virus structure. That would have been first prize, is just synthesize some virus and just look at that, because then you have a complete, much more in situ situation. But unfortunately, you can't really transport live virus without a lot of regulatory hurdles that you have to cross first. So I was stuck with recombinant protein. How I would look at the flexibility and whether or not it has a function. There's some speculation about the virus having tropisms switching between different cell types, either in the same host or between the midge and the horse. So when it's in the horse, the VP2 needs a particular structure, and it's in the midge, it might need a different structure in order to bind a different cell type. And that makes sense because they're quite different creatures, evolutionarily speaking. So there might be some conformational changes in the tip in order to have host switching being possible. There's also the idea of serum cleavage or proteolytic cleavage in the saliva of the midge that assists in this host switching. So there's possibility that the flexibility is functional. How I would test that, it would be nice to figure out what receptors it binds in the different hosts, and then see if you treat it with certain proteases, see how it then enhances or reduces the tropism for particular cell types. And if you can even combine that with the structure, then you can see how, look, this conformation is enhancing the binding here after treating with this protease, and after treating with a different protease, it's binding this receptor, and they can change shape thanks to this flexible component. The reason I say it's flexible is because there aren't any secondary structures. And if you look at the different alternative models that AlphaFold gives you, it actually moves the tip domain around this flexible hinge. So it's quite likely that the flexibility exists there in that hinge, and it might have a function.
Hi, can I ask a question? Sorry, I don't know how to put up my hand in this new version of Zoom. Kind of just if you go back to your image, kind of your final structure, these two, the hairpin and that helix tone helix motif, you say that those might both be interacting with VP5. I mean, I don't know that much detail about the arrangement on the virus surface, but these two elements seem to me to be lying on opposite sides of the protein. Is the arrangement such that both of them could nevertheless interact with VP5? Or is it, yeah?
They are quite close together, the VP2 and VP5.
Yes.
And just looking at them, the two full structures, it's difficult to really see how close they are with the 14 angstrom wool sickness version, but I would say it's in a plausible distance, because they already implicated the loop in a VP5 attachment, or rather VP5
Which one,
VP2
the
interaction.
helix, or
The hairpin loop.
the hairpin and blue tongue virus. Okay.
So missing the hairpin loop, the only alternative would then be the helix loop helix to be an alternative way of stabilizing the VP5 VP2 interaction.
But you could possibly put this, or kind of, I mean, in the blue tongue, you've got the whole virus, so there you could really check whether that hairpin is associated with VP5. But then for the African horse sickness virus, you might also be able to reconstitute the virus particle, roughly at least, and just check how far it is from VP5.
Yes, I could try and dock them. I think African horse sickness VP2 is the base domain to VP7 is quite close in structure, so I can try and dock it in the electron density of a higher resolution.
Yeah, just something like that, just to check that that's actually,
see what the interaction distances might be.
Okay, sorry, I think you're cutting out, or I'm cutting kind of the connection.
Yes, that's a good idea. I saw someone's hand.
I have another question if there's time.
It's probably the weather.
Trevor, if there's time, I have another question.
Go ahead, Lawrence.
So I just wanted to go back to Henk's thing about the transporting virus particles for structural biology work. Trevor, could you or Henk, could you talk about what's involved in that? What are the approvals and what are the impediments of whole virus structural biology?
I see Albie is here. She can perhaps explain the law better than any of us.
Yeah, Trevor, not really. Spotty would be the better one to be here, but I think there was a structure of African horse sickness virus 7. The one which has a deletion in VP2. It would be very interesting to sort of compare your full-length VP2 structure to that VP2 which has a part which is deleted. It will be very interesting. So I would really, I think it's African horse sickness virus serotype 7 with the truncated VP2. I think it's important that you see how your structure of serotype 4 maps to that one. That's the most genuine, how you can put it into the real particle. Because I do think also that in terms of African horse sickness and causing disease, there's something which is called serotypes, and there are 9 African horse sickness virus serotypes. And the serotypes are mostly defined by the outer capsid proteins, specifically VP2, which gives rise to neutralizing antibodies, and the neutralizing antibodies correlate with protection. So this whole business about which part of VP2 sits where, and specifically the tip, as Henk referred to, that tip has a lot of secrets which we are only now starting to be able to appreciate now that we can do reverse genetics. So you can do a lot of reverse genetics and make some changes and then new structures and see how that affects the structure, and then also back to the biology in the virus and the infectivity in cell culture would be very interesting, and neutralizing antibodies. There's a lot of neutralizing antibodies available. So I think you've got fantastic work, very well done. So can you just come back to this comparison, if you had a look at the comparison to the truncated VP4 structure of African horse sickness?
I looked at the truncated version, and This is a VP7. So this got truncated in cell culture through the attenuation process. So it became more infectious to the cell type that they were culturing it in, as you expect natural selection to naturally select. And one of the features of the VP2 in the truncated version is that the majority of the tip domain just isn't there. So it got sort of removed. It's likely that whatever the conformational change that happens, either through proteolysis or some sort of ion detection, it's possible that the opening up of VP2 is necessary prior to infection, and how that happens might be the next thing to study. I think having neutralizing antibodies binding to VP2 might also even help with the orientation problem, because if you add some bulk to it, if you can shield some hydrophobic regions, you might prevent it from only taking one orientation. So that's one of my plans is to maybe see if I can resolve the structure of antibody bound to VP2. There's also other ideas to get around the orientation problem. First prize would be being able to just look at the full virus, but yeah, that's another thing I'll try and look into. But the truncated serotype 7 is a really interesting thing that's sort of guiding everything I'm planning in the background. Yeah.
There's a question in the chat from Mamaru. Mamaru, do you want to ask your question? Mamaru, are you still here? Oh, yes, you are there.
Yes,
you-
yes.
Yeah.
Henk, it was very nice talk, and I already wrote my question in the chat box, and so somewhere in your slide you added zinc ion, and if I understand it correctly, naturally zinc ion doesn't exist in the protein structure of the virus you are studying. So why you specifically add zinc ion? Can you explain it, please?
Okay, so in the protein, there's this domain called a zinc finger domain, and it actually chelates zinc. Obviously, zinc isn't part of the codon table that a cell can use to build a protein, but you can build these domains that form these chelation sites, and that's where zinc just spontaneously will bind and stay stuck in. And in VP2, it's suspected that it's there for stability, and it actually maybe came from a DNA binding protein, and it stayed there just because it's really good for the stability of the protein. The reason I had to manually add it is just because Rosetta didn't have ligand support. So this is a ligand that should be there, and it is there in the correctly folded protein, or we suspect it to be there. There's electron density that supports the location of the zinc ion. But the way I added it manually is just the technicality rather than actually related to the protein or the experimental process. I'm also experimenting with zinc concentrations to see if that maybe is responsible for some of the instability in certain parts of the protein. But yeah, the zinc is actually there.
Thanks, Henk. Are there any further questions? I can't see. Well, nobody has their hand up.
So Trevor, is this a compelling piece of work to talk specifically about why South Africa should invest more in structural biology?
Absolutely. Albie has been aware of this forever.
Yeah.
And so the problems are that getting this done outside of the country is much harder than it is theoretically getting it done inside the country if there were equipment available. So, and this applies to all viruses, especially infectious plant and animal viruses that are perhaps more tractable and more localized than human viruses. So it is certainly something that we have to emphasize when asking for resources of this of the CryoEM nature in Africa. Okay, I think with that, I think I'm going to wrap up the meeting, unless anybody has any further comments or questions.
Nope, that's it.
Okay, then thank you very much for coming. And thank you again to Henk for an excellent talk and a very good exposition of a very difficult cryo-EM problem. Thank you.
Thank you.